All right, welcome to Chapter 6, Genetic and Developmental Disorders. This is another very vocabulary and concept-heavy chapter. Luckily, it should be mostly a review or very familiar from A and P. So I very much suspect you'll have to go through and review genetic stuff again, but this will serve as a good review, hopefully, and you can supplement where you need to. All right, so we're jumping right in with DNA mutation and repair. So there are different kinds of mechanisms that go hand in hand here, and different things that can happen. So a single gene, which is also a Mendelian, I think is how you say it, disorder, is caused by DNA mutation that codes for a particular protein. Chromosome structures are also mutated through loss, gain, or translocation of segments. So again, this is a good spot for you to review the different ways that we can have our DNA mutated. Mutation. This is a permanent change in DNA sequence or chromosome structure. It's pretty rare. Potential mutagens are radiation, chemicals, and viruses. May occur spontaneously as a result of DNA copying errors. Mutagens may cause single-stranded breaks. These are easily repaired and they only affect a single gene. Double-stranded breaks are possible permanent loss of genetic information at the breaking point, and this affects a single gene or a whole chromosome. And here is a video that does a really good job animating that for you.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base or bases are added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll get a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. While unfortunately there is not a cure for this disorder yet, the good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So I really like their animations. If you also like them, maybe check out their channel because they have some really good stuff. So that explains a lot about mutations, which I thought was a good review. All right, so now we're going to look at genetic disorders versus environmentally induced congenital disorders. So genetic disorders, these are apparent at birth or appear in later life. Majority are inherited from the parents and some from embryonic development mutations. They're divided into groups. So they're chromosomal aberrations, the single gene disorders, multifactorial polygenic disorders, single gene but does not follow the Mendelian pattern. So we have triplet repeat mutations, mitochondrial mutations, and mutations affected by genomic imprinting. So congenital disorders, and or malformations are present at birth, genetic or environment or both, and they are associated with structural deficits caused by errors in fetal development. So here's another video that kind of explains a lot more about that.
Hi, in this video, we're going to talk about chromosomal abnormalities and non-Mendelian patterns of inheritance. Now, we've already mentioned one type of abnormality that can occur, and that is non-disjunction, when sister chromatids or homologs fail to separate during cell division, specifically during meiosis. Now, what you'll wind up with, if you have a non-disjunction, you'll have an extra chromosome going to, let me take myself out of the image here so you can see this. You'll have an extra chromosome going to one cell and another cell missing a chromosome. And this results in aneuploidy, having the wrong number of chromosomes. Now, aneuploidy can result from other things, but it does commonly result from non-disjunction. And when you have aneuploidy, you can end up with organisms that have monosomy, having only one chromosome instead of the normal two, or you can have trisomy, which is 3 chromosomes instead of the normal 2. And a very common example of trisomy is Down syndrome in humans. And Down syndrome results from trisomy of the 21st chromosome. And there are a number of symptoms that go along with this. Now, some organisms are polyploidy, meaning they have more than 2 complete sets of chromosomes. Plants actually can be triploid or tetraploid, having 3 or 4 complete sets. And plants actually do all right with these extra chromosomes, but it's pretty harmful to humans and other animals. We don't do well if there is an upset in the number of chromosomes we're supposed to have. Now, there are other types of abnormalities that can occur, and these come in the form of chromosome damage and botched repair. So here we have a normal chromosome, and sometimes what you can end up with is a deletion where part of that chromosome actually is deleted and removed, and then you have a truncated chromosome or a shortened chromosome that's missing a piece. You can also have duplications where an extra fragment of a chromosome will be duplicated and attached to that chromosome. This is, in some ways, this can be just as bad as a deletion. It can be just as harmful. Now, when you have a duplication, sometimes that results from a fragment breaking off and being reattached to the wrong chromosome, but that fragment can also be reattached in the wrong orientation, and we call this inversion, where you actually have that fragment, but it gets reattached backwards, as we see here. This is very dangerous, as genes need to be read in a specific direction. So this is going to cause all the genes to be read backwards, and it's going to be very harmful to the organism. You can also have a translocation where a chromosome fragment from one chromosome actually attaches to a non-homologous chromosome, attaches to the wrong chromosome. And this also can be quite damaging. Now, let's turn the page.
All right, so again, kind of a different take on some of the same concepts from the other video, but I feel like these visual representations are really helpful sometimes just to kind of understand all of this content that is very heavy in some pretty complicated concepts. All right, So the causes of all of this happening are poorly understood, but we have identified some risk factors. Advanced maternal age can lead to some of these chromosomal abnormalities, abnormalities in parental genetic structure, and abnormalities in crossing over. All right, so this usually resulting from breakage or loss or rearrangement of chromosomal pieces during meiosis or mitosis, as we just saw. Meiosis, this is where we have the crossing over error. So chromosome portions are lost, attached upside down, or attached to the wrong chromosome. And in mitosis, opportunities for chromosomal breakage and rearrangement. And he mentioned trisomy 21, which is Down syndrome. This is the most common chromosomal disorder, the leading cause of mental retardation, and it is clearly associated with advanced maternal age. Again, that is a risk factor, though, it does not cause it. And here are some of the characteristics we see classically presented with Down syndrome or trisomy 21. This is here for your reference. We don't need to go over all of them. Let's see. So here are some other examples. So trisomy 18 is Edwards syndrome, and trisomy 13 is Patau syndrome. So these are much less common than trisomy 21. They result in severe mental retardation. Most pregnancies are lost before term, and infants usually die a few weeks after birth. So trisomies 8, 9, and 22 are very rare, but they happen. And it looks like that. Oh, it is not the end. I thought it was the end. Okay, so hold on. Let me get my place, because we definitely skipped forward there. Hold please. Oh, there we go. Okay. Cray-Duchat syndrome. So this is deletion of the short arm of chromosome 5. It results in severe mental retardation, a round face, congenital heart anomalies. The cry resembles a cat crying, and some live to adulthood and thrive better than those with trisomies. There is a picture of this in your text. Klinefelter syndrome. So this is the most common sex chromosome abnormality. It affects males only. Usually one extra X chromosome, sometimes more than one. Abnormal sexual development and feminization, and a lack of secondary sex characteristics during puberty. There's also a picture of this in your text. Turner syndrome, 1 in 250 or 2,500 live female births, so it's not all that uncommon. This is monosomy X, so they get one normal X chromosome with no Y chromosome. The female phenotype with no developed ovaries, the second X chromosome missing or structurally abnormal. This is usually from the father's chromosome. So these single gene disorders, they result from alterations or mutations of single genes. Affected genes may code for abnormal enzymes, structural or regulatory proteins, or regulatory RNA. The classification for these is the location, so autosomal or sex chromosome, and the mode of transmission is dominant or recessive. So here is a pedigree chart. We're looking at an autosomal dominant disorder. So this is caused by mutation of a specific autosomal gene. Males and females are equally affected. Usually one affected parent. The unaffected individuals do not transmit the disease. And the offspring of one affected parent with an unaffected mate has a 50% chance of inheritance. And the offspring of two affected parents, 3 and 4, which is a 75% chance. So you see that here, the top one, this top graphic here shows one affected parent, one not affected parent. And as you can see, 50% chance. When we have two affected parents, we end up with a 75% chance. So Huntington disease. This primarily affects neurological function. Symptoms appear after age 40. There's mental deterioration, involuntary arm and leg movements, and the parent may transmit to offspring before becoming aware of the defective gene because symptoms don't typically appear until after age 40, which many individuals have children prior to age 40. So mutation of autosomal recessive genes, males and females are equally affected. Usually not apparent in the affected individual's parents. Both parents are carriers of a mutant recessive gene. So the unaffected individuals may transmit to offspring. So if you have two carriers have 1 in 4 chance of having the affected offspring and 2 in 4 chance of having carrier offspring. So here are some autosomal recessive disorders. Albinism, this is the lack of pigmentation from disrupted melanin synthesis. And then also PKU. So this is an inborn error of metabolism. They cannot metabolize this specific phenylalanine. And so this is tested for at birth. So testing for PKU may be something you've heard of, or it's definitely something you will hear of in the near future. Cystic fibrosis is another autosomal recessive disorder, and it's the most common genetic disorder in people of Caucasian descent. The production of abnormally thick secretions in glandular tissues from a defect in chloride transport across the cell membrane. And they have a reduced life expectancy and frequent pulmonary infections because they have thick secretions in their lungs and are not able to get bacteria and whatnot out of their lungs as easily because those secretions are thick, so they don't come out. Sex-linked or X-linked disorders. These are a mutation of the sex chromosome, almost always the X. Nearly all X-linked disorders are recessive. They are almost always male. Males always express the disease and they only have one X chromosome. So the affected fathers transmit the defective gene to all daughters, but to none of their sons. The unaffected males do not carry the defective gene. So carrier female has a 1 in 2 chance of producing an affected son or carrier daughter. Females are rarely affected. These are homozygous state of an affected or carrier mother, X-affected father. Hemophilia A is an example of this, and this is a bleeding disorder from the lack of a factor. It's factor 8. All right, so on to environmentally induced congenital disorders. So these are errors in fetal development that result in congenital malformations. So this is not an issue with the way that cells are acquired and dividing from the mother and father. So that genetic piece of it, this is something happens environmentally that disrupts normal function of the cells as they're dividing. So these are errors in fetal development that result in congenital malformations. Teratology is the study of developmental anomalies. And teratogens are factors that cause congenital malformations. So these can be chemicals, radiation, viral infections. So these are the things that a lot of times they are keeping an eye out when you have an expectant mother, and we're cautioning, educating, that kind of thing. So the susceptibility depends on the amount of exposure, developmental stage of the fetus when exposed, prior condition of the mother, and the genetic predisposition of the fetus. Lots of factors there. So these are some general periods of fetal vulnerability. Before the 3rd gestational week, teratogens exposure either damages very few cells or so many that the embryo cannot survive. From the 3rd to the 9th week, the embryo is very susceptible to this, especially in the 4th and 5th weeks during organ development. And after the 3rd month, it can affect a growth or injury to already formed organs. So teratogen, this is a difficult word for me apparently. Teratogenic agents, they interfere with cell proliferation, migration, and differentiation. Chemicals and drugs include alcohol and thalidomide, and infectious agents or viruses. And here is a little acronym to remember the most important ones. So toxoplasmosis, others, rubella, cytomegalovirus, megalovirus, and herpes. Radiation is also a concern here. So other infancy disorders, they generally arise later in uterine life. They usually involve the health of the mother or the placenta, and they result in low birth weight, immaturity, and fetal brain injury. And that is the end of this lecture and the end of week 1. So congrats on making it through all the lectures. Please reach out if you have any questions or need any clarification. Your book has a plethora of information in it. You can also look for supplemental videos on YouTube. That's what I typically do when I'm trying to learn something new or understand something, because I'm very much a visual learner. I have a harder time reading and absorbing personally. So utilize those resources and reach out if you need anything.